sorry, it's over air now. It's still nothing's happening here. So it's totally blank and the,
Okay, so we went through the questions from yesterday. So now, today we'll talk about, um, well, remember yesterday we talked about the hormones. Now we're going to talk about the receptors. And we'll talk about the general mechanisms of action, how each receptor works. We'll talk about how receptor signaling is finished. How do you terminate the signal? And we talk about how you measure hormones, these different hormones we talked about. So we'll first discuss the different types of receptors. We'll talk about steroid hormone receptors, GPCRs, enzymatic associated, so tyrosine kinases, and then the quantification that I mentioned to you. And remember, when we talk about our Nihao hormone, right? The only person that receives the hormone, the only cell that receives it, is the person with the right receptor, okay? So the target cells have to have the right receptor, okay? They're only in our QQ group or they're only on our WeChat. That's the receptor. So how does this work? How do you get a specific hormone to bind to a specific receptor? Usually, we talk about a lock and key mechanism. So, very similar to medicines, hormones produce their effects by interaction with receptors, which may be intracellular or membrane bound, depending. The intracellular receptors are the steroid hormones because remember, the steroids can cross the plasma membrane. The membrane-bound receptors are usually G protein coupled receptors or enzymatic enzyme-associated receptors, the tyrosine kinases. Okay, so, and they have this lock and key where the specific ligand binds to the receptor. So how do these steroid hormone receptors work? You have your steroid hormone here in yellow, and because it's able to cross through the plasma membrane, all the way across to the cytoplasm, it reaches the nucleus where the steroid hormone receptor is located. It will bind to this receptor, and then usually it can act as a transcription factor and bind DNA and immediately impact transcription and translation. And remember, we talked about the timing of effects, right? Because it can diffuse readily across the phospholipid bilayer, and it can bind and activate these intracellular receptors and modify transcription and translation. The timing of these receptors and the mechanism is very different than the GPCRs. So after it translocates and interacts with the DNA, it triggers this gene transcription and produces messenger RNA. And because that process is very slow, you have to make more message and make more protein. there is a slow onset of action compared to the GPCR, but it is more prolonged, right? It's going to last longer. So what about GPCRs and enzyme-coupled receptors? GPCRs are on top here. They are 7 transmembranes, and a lot of this should be reviewed from the pharmacology. They are coupled with a heterotrimeric G protein complex shown here in green. After the hormone binds the GPCR, it leads to a conformational change, okay, which exchanges GDP for GTP on the trimeric G protein complex. And then this complex can go and activate an enzyme. Remember, these indirectly regulate a target protein, okay? Unlike the steroid hormones where it's direct. And they mediate their action via this trimeric GTP complex. For the enzyme-coupled receptors, usually their hormones act as dimers. This causes dimerization of these receptors. They have an active catalytic domain, which usually cross-phosphorylates or self-phosphorylates. And then this goes on to drive a signaling cascade. Usually, these enzyme-coupled receptors function as enzymes or directly associate with enzymes, and they are heterogeneous in structure. So, just a little bit about G protein coupled receptors. This was the Nobel Prize from a few years ago from a man named Professor Brian Kobielka and Professor Lefkowitz. Professor Kobielka has an institute that he works with in both Beijing and in Shenzhen. These are some of the important medicines that are targeting G protein coupled receptors. And this slide is now a few years old, but still you see GPCRs are very, very important. In fact, some of you may know there's a professor in Nanchang, very famous for studying GPCRs. In English, we call him Jin Zhang. I don't know. I forget what the Chinese name of Jin is, but he's a fantastic professor there at the medical school in Nanchang studying GPCRs. More than 50% of the medicines people take today treat a broad spectrum of diseases by targeting a few G protein coupled receptors. So these include antihistamines, bronchodilators, beta blockers, blood pressure, targeting drugs, pain, and migraine. 40% of the top 200 drugs act on GPCR. So these receptors are very, very important. There are still many, many GPCRs that need to be understood, with over 600. But what is interesting about GPCRs is they all look very, very similar, right? They all look like this. So they have 7 transmembrane domains. Typically, glycosylation groups on the outside. They are phosphorylated in the intracellular side. So how do they work? So drugs that target these receptors, and even some hormones, act by either binding to the receptor and causing a full agonist response. Now, some of this should be reviewed from your pharmacology from Choi. Partial agonist response, right? A neutral antagonist, something that blocks, and that's many medicines, or an inverse agonist. All of these can be drugs, or all of these can be mechanisms of action. So how does this work? So receptors are like dancers, right? You know, when you go in the parks, right, and you have the people, some of the old ladies, and then sang dancing to the wonderful music, right, for exercise. They're moving, right? Always different poses, right? Sometimes some doing Tai Chi. So their receptors do the same thing. They're always moving, okay? So each one of these motions, this motion, you can plot in an energy diagram. If you remember your basic chemistry and physics, you have conformations. This is the dancing poses, and you can be an inactive receptor or an active receptor, and this is the energy. And usually, remember, everything likes to be at the lowest energy state, so it'll be in an inactive conformation. And to get active, you put energy into the system. Well, the cell does this with the receptor by adding G proteins. So if you add a G protein, which is this MB80, and you add a hormone or a drug, you move this active state lower. So now the lowest energy state is the active conformation. And that is because we have the hormone and our G protein. Okay? So what I want you to remember is that receptors are always in motion, and they try to obtain their lowest energy state, which the hormones and the G proteins, in the case of GPCRs, lets them achieve. And this motion is directly linked to their activation. So, in the case of G protein coupled receptors, what does this activation lead to? When the hormone binds to the receptor, it causes a second messenger production of a variety of second messenger molecules. This can be inositol triphosphate or cyclic AMP. And these go on and lead to the physiologic changes that we would expect. So, the cycle is, first, you have our receptor and you have our heterotrimeric complex bound to GDP. When an agonist binds to the receptor or a hormone binds to the receptor, you have an exchange of GTP for GDP. This alpha GTP separates from the receptor and from beta gamma and can then go activate an effector. And beta gamma is understood to now go on and also interact with other effector channels. Once the ligand then releases from the receptor, this complex can come back together. GTP gets hydrolyzed, and you now have the whole system reset, ready for another cycle. So, what are some examples of hormones that act via GPCRs? We have many of our releasing hormones, right? Remember, the releasing hormones come out of the hypothalamus. We have some of our pituitary hormones. Okay, just a few lists. So one of my favorite questions is, tell me a hormone that binds to GPCR and tell me how it works and how it signals and what the physiological consequences are. What's the downstream target? So to get different second messengers activated, the receptors use different G-alpha proteins. So we have G-alpha S, which stimulates adenylate cyclase to increase cyclic AMP. We have G-alpha I-O family, which inhibits cyclic AMP production. G-alpha Q, which stimulates phospholipase C, leading to an increase in IP3, that inositol triphosphate. And we have the G-alpha 12-13 family, which stimulates RhoGEF to activate RhoA. And this is just a video, an image of those pathways. So what are the proteins responsible for activating and amplifying a signal after a GPCR is activated? It is these heterochimeric proteins that we mentioned, but importantly, this important step of GDP to GTP exchange causes a conformational change in alpha, which is driven by conformational change that we talked about within the receptor. This alpha GTP is inherently unstable, and once it hydrolyzes, the GTP will go back to an unactive GPCR. Okay? And that's again highlighted here. One of the things I didn't mention earlier was these myristyl groups on the G protein to help recruit to the receptor. The downstream implications may look familiar from the standpoint of the steroid. Remember, with the steroid hormones, it went straight to the nucleus and bound to the DNA. In this case, we'll activate adenylate cyclase, produce cyclic AMP, and then go in and drive transcription and translation. But unlike the steroid hormones, we require this second messenger production, okay, and then binding of the DNA. Okay, the gene transcription is driven not directly, but indirectly. Now, an important kinase that's part of this cyclic AMP cycles, if you will, is protein kinase A. Protein kinase A has 4 subunits. 2 of these will bind to cyclic AMP when cyclic AMP is produced by adenylate cyclase. Cyclic AMP binds these regulatory subunits. It releases these inactive subunits to now become active, and these catalytic subunits can go on and phosphorylate a variety of intracellular targets. Sorry, my music's not working. So for any Star Wars fans out there, my children love Star Wars. The complex of adenylate cyclase and the protein kinase A looks very similar to R2D2. Cyclic AMP binds this regulatory subunit that I mentioned, and then you go on and get this active protein phosphorylation. Now, cyclic AMP and pKa are not the only pathways, so we talked about GQ, alpha GQ. Alpha GQ leads to an activated PLC beta, which then, in this case, interacts with lipids at the membrane. It hydrolyzes this PIP2 into this PIP IP3 complex. And eventually this diacylglycerol is another byproduct of that reaction. This leads to an increase in intracellular calcium and eventually an activated PKC complex. So GQ leads to IP3. Calcium signals are an important second messenger of the hormones that we're talking about, along with cyclic AMP. Calcium is sensed in the cell by calcium binding proteins, some of which include the calmodulin family. Calmodulin works by binding, very similar to what I showed you before. It has a regulatory domain that binds calcium. This changes the conformation of the receptor, can drive activation, and which then hydrolyzes ATP. So that's steroid receptors and GPCRs. What about enzyme receptors? This is the enzyme receptors below. The enzyme coupled receptor class is very large. It includes receptor tyrosine kinases, tyrosine kinase associated receptors. The receptor tyrosine kinases phosphorylate tyrosines on themselves and then the other targets, whereas the tyrosine kinase associated receptors have no intrinsic enzyme activity, but their role is to recruit kinases that then phosphorylate and relay the signals. Then you have receptor serine threonine kinases, which do directly phosphorylate specific serines or threonines on themselves. Histidine kinase associated receptors are also in this family, which activate a two-component signaling pathway in which the kinase phosphorylates itself and then transfers this phosphoryl group to an intracellular signaling pathway. You have the receptor guanylyl cyclases, which catalyze the production of cGMP, which is very similar to cAMP. And then you have the receptor-like tyrosine phosphatases, which remove phosphate groups from intracellular tyrosine. The key feature, okay, and again, a very popular question that I ask, the key feature of receptor tyrosine kinases or enzyme linked is this dimerization. Okay, so they're inactive when they're single transmembrane domains, and then the hormone drives dimerization. You get cross-phosphorylation and eventually a nice signaling cascade after recruitment with a variety of other intracellular signaling proteins. We have this trans-autophosphorylation. What does this do? It drives recruitment of other enzymes. An example is this activated PI3 kinase, which also can start using that PIP2 that we mentioned before and and phosphorylate to make PIP3. This then recruits a series of pH domains that bind specifically to PIP3 containing lipid rafts, PDK1 and AKT, perhaps the most famous. This can then recruit other important regulatory mechanisms like mTOR, which helps drive transcription and cell division. And then eventually things like apoptosis, which are all cancer related. So you'll hear about some of this with Richard Gross when you start studying the cancer module. This is an example of the tyrosine kinase receptors. Okay, it's NGF receptor. You can see here this ligand is help driving dimerization, and you've got a variety of these domains in the single transmembrane domain with the active kinase domains on the intracellular side. In terms of enzymology, endocrinology, you have the insulin receptors and some of the growth factors, EGF, PDGF, IGF, and NGF. In this case, one hormone molecule is binding to each of the receptors that then leads to dimerization. Dimerization allows the two cytoplasmic domains to phosphorylate each other because they come together, and then you get this downstream activation. Now, this is the case where they're single polypeptides, but sometimes the receptors can act with multiple polypeptides. So in this case, you've got insulin receptor. It's got this cysteine-rich domain. You see it's two different polypeptides linked by a disulfide. And the structure looks like a nice arch, right? Nature is very beautiful. Look at this. Looks like architecture. But you can see how binding here can bring together these kinase domains at the bottom. And this is a little video. Let's see if it's going to work for me. Sometimes it works, sometimes it doesn't. There we go.
Cell membrane, so it has a part outside, a part inside, and insulin binds to the part that is outside. Insulin is a small molecule whose structure was determined a long time ago, in 1969. But it's been unknown how it interacts with the receptor, and that is what we have discovered. And it's interacting.
It's not working, the sound.
The sound is working, but it's so mild, so it doesn't hear clearly. Clearly here.
So it's too loud.
It's too slow. I mean too small.
Okay, so maybe I turn the volume down and just watch the video. We try that?
No one can hear it. You can't hear it.
Yeah, I turned the volume down. Is that better?
No. Can you just turn it upwards? Turn it
The two
louder.
parts inside of the cell unwind and make contact. These two proteins then activate each other.
Can you see the video?
We can see the video, but the sound is totally mild.
Okay, don't worry. The sound's not important. What I want you to see, you see this dimerization down below, and now that they're activated, they cross-phosphorylate, right? Can you see that? That's the intracellular domains. And now it's recruiting other kinases, right? These other red proteins are these are the proteins that will then drive the interaction and drive the signaling. Okay, so now those are activated and they'll drive the signaling cascade and start phosphorylating other proteins. You can see that that's them recruiting, dimerizing, cross-phosphorylation, and then they can lead to the signaling cascade that is downstream. Okay, and then on the right, you can see part of the problem if you have a non-active type insulin receptor, then you won't get the signaling cascade, and that's partly what drives diabetes. Now, some receptors of cytokines and growth factors that drive cell proliferation and differentiation. This becomes important, for example, in development or in some of the tumors that we'll talk about, or in regeneration of, say, the adrenal gland. They do not have intrinsic tyrosine kinase activity, but they can associate with non-receptor tyrosine kinases. So this is your JAK-STAT proteins. So these receptors recruit these enzymes, these kinases, and then that would lead to downstream signaling. Some examples from for us, for endocrinology, will be growth hormone, prolactin, leptin, GM-CSF, and some of the important downstream signals are these JAK kinases and SRC. Still, ligand binding can result in receptor dimerization, which leads to activation of these non-receptor kinases. So you may have understood that there's a lot of cross interaction between GPCRs and receptor tyrosine kinases. So some of these very similar pathways overlap with each other, right? We talked about phospholipase C, we talked about diacylglycerol, we talked about calcium and IP3, PKA, PKC, AKT. A lot of these are very similar pathways, so they can really interact with each other depending on the hormones. And if you think back to what we talked about for endocrinology in terms of regulation and feedback, this is partly how these feedback mechanisms are achieved, by shared signaling pathways. Okay, why don't we take a pause here, and then we can come back in 30 minutes and we finish up, okay?